Now, here's the opportunity to practice, to put everything together, kind of join all the information we just went over together, and we're going to now we're going to do transcription first and then translation. First thing we're going to do, so DNA, copy DNA. So we're going to make an opposing strand of DNA. And what you would do, here's a section of DNA. This could be a gene. Genes are, I think the shortest human gene is about 1,000 of these, but this one's pretty short. So first thing, you're going to fill in what is called the opposite corresponding nitrogenous base. So ATGC, adenine, thymine, guanine, cytosine. So you want to fill in on the opposite side the corresponding base pairing. A's always bind with T's, and T's bind with A's, vice versa. G's always bind with C's, and then C's always bind with G's. So what you would do, so I'm going to put a T right there opposite the A. So adenine binds to thymine. Then put another A. Opposite the G, guanine. Of course, that's going to bind to cytosine. Opposite the C, you're going to put a G, if you see how this is going. So one more, and then adenine. Of course, you're going to put a T binding to thymine, and then fill out the entire DNA strand. Ooh. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. In 3, 2, 1. Okay, we have now filled out the entire strand, and if you count each base pair, there should be 24 on the top, 24 on the bottom, and we filled in the corresponding DNA. You match it with the corresponding nitrogenous base. So next, we're going to do transcription. So what that means is this section of DNA, we now want to make a strip of messenger RNA. Remember, RNA has 3 base pairs that are the same, but instead of thymine, RNA, it gets replaced in RNA with uracil. The reason that is a cell synthesizes RNA, and thymine costs more metabolically to make than uracil, and uracil still accomplishes the same thing. So think of uracil being like the off-brand. So what we would do, put an A opposite the T, and now opposite this A, where you would normally in DNA put a thymine, you're going to put a uracil. Opposite the C would be a G, and then you fill in the next few. G would be C, and then opposite the T, you're going to put an A. Then opposite the A, you're going to put another U. And then fill in the corresponding messenger RNA. Well, you fill in the corresponding bases, but you're going to make a messenger RNA strand, just like it says right here. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24. In 3, 2, 1. All right, now we have finished filling in our messenger RNA strand. If you count each base pair, in our particular example, it should be 24. Now, these represent the transfer RNA molecules that are going to move out into the cell cytoplasm and attach to the correct amino acid. And so what happens is this messenger RNA strand is going to go out and attach to a ribosome, and the ribosome is going to start translating the genetic code, which these are in threes, they're called codons. And these transfer RNAs would then go out and attach to, depending on what this code says, the codon, the triplet, the correct amino acid. So that's where this chart comes in. Now, starting off, this is methionine. In 3, 2, 1. So you have a start codon, this MET, methionine, or methionine. I've heard it twice, both ways. See the AUG? Okay, that transfer RNA is going to go out and attach to that. So you can put here MET, could have put start, CAU, going back to our chart, too much caffeine, CAU, there it is, histidine, HIS, so it will bring it back to the site of the ribosome, and it's going to drop the amino acid in the sequence. These are transfer RNAs. So we're gradually building what is called a polypeptide. We're making a protein. The next three, CUG, CUG, leucine, CUG, yeah, LEU, wrong button. There we go. What are the next three? And you would do this for the entire thing. So you would then go in and fill out the corresponding, put the correct amino acid depending on what the codon is. All right, I have now finished filling in the corresponding, the amino acids. These were transfer RNAs, and they went out and grabbed all of these amino acids, and that's translation. This was the start codon, and see this UAA? There are three of these, UAA, UAG, and UGA. They represent what are called stop codons. That's how the ribosome knows to stop reading the messenger RNA strand. And now you have a fully functioning protein. Ours is really not that big, of course, but you think about a protein like hemoglobin. Hemoglobin is 256 amino acids. So we did 1, 2, 3, 4, 5, 6, 7, 8. So ours is pretty small. I went ahead and spaced these out so they would match up with the transfer RNAs, and I did the lowercase just like in our chart. But that is transcription and translation in a nutshell.
Command that Nedry used. He's covered his tracks far too well. And I think it's obvious now that he's not coming back. So, shutting down the entire system.
You can get somebody else because I won't do it.
Shutting down the system is the only way to wipe out everything that he did. Now, as I understand it, all the systems will then come back on their original startup mode. Correct?
Theoretically, yes, but we've never shut down the entire system before. It may not come back on at all.
Will we get the phones back?
Yes, again, in theory.
What about the lysine contingency? We could put that into effect.
What's that?
That is absolutely out of the question.
The lysine contingency is intended to prevent the spread of the animals in case they ever get off the island. Dr. Wu inserted a gene that creates a single faulty enzyme in protein metabolism. The animals can't manufacture the amino acid lysine. Unless they're completely supplied with lysine by us, they slip into a coma and die.
People are dying.